This is the third of a four-part lecture series for Lecture 19, the topic of mutations. And in this part, we're going to consider mutations that can occur as a result of mutagenic agents, specifically chemicals and radiation. In the previous lecture, we talked about the causes of spontaneous or naturally occurring mutations. So induced mutations are those that increase the rate of mutation above the spontaneous background. They are mutations that are as a result of exposure to mutagens, and mutagens are physical and chemical agents that induce mutation. Some mutagens are naturally occurring. For example, fungal toxins like aflatoxin, found in different types of Aspergillus fungi. It's a concern in moldy peanuts. Ultraviolet light is a naturally occurring mutagen. Polyaromatic hydrocarbons, or PAHs, are mutagenic compounds that are formed whenever you burn organic materials. So whenever you have a forest fire, for example, you generate PAHs. But also when you burn cigarettes, you generate PAHs. And then there are many mutagens that are really expressly due to human activity. For example, radiation from nuclear bombs or X-rays, or the PAH that are present in cigarette smoke, or various industrial pollutants. So there are many different kinds of mutagenic agents. We're going to talk about base analogs, alkylating, intercalating, and adduct forming agents, ultraviolet light, and ionizing radiation. Base analogs are compounds that can substitute for nucleotides during DNA synthesis. For example, shown on the top, we have thymine, which is a normal, naturally occurring nucleotide base. And we also have a chemical called 5-bromouracil in both its keto and its enol form, so the two tautomers of 5-bromouracil. 5-bromouracil is not a normal, naturally occurring base. It is a base analog. Okay, so 5-BU is a base analog. When 5-BU is in its keto form, it will base pair with adenine. So when it's in that form, it is behaving like naturally occurring thymine. When 5-BU is in its enol form, it will base pair with guanine. So not acting like naturally occurring thymine at all. So if you're growing cells and you put 5-bromouracil into the growth environment, it will be substituted in as thymine, and it will behave either as thymine or instead like something that will base pair guanine, a little bit more like cytosine, for example. So it will cause a mutation at that one position when it is present in the enol form. Now, if you encounter a base analog in nature, then that could be a problem, and you could have a mutation event in your DNA. You might choose to use base analogs in the lab to purposefully induce mutations in organisms that you're studying. Or another thing to think about is that some base analogs are actually used therapeutically. So shown here on the top right is a picture of AZT, also called zidovudine, or another chemical name for that is azidothiamidine. This is in fact the first anti-HIV drug developed. It is sold as something called Retrover, and it is prescribed for the treatment of human immunodeficiency virus infection, HIV infection. Its mechanism is that it is an inhibitor inhibits reverse transcriptase. Reverse transcriptase is the step that HIV, which is a retrovirus, uses in its life cycle when it takes viral RNA and it converts it to viral DNA. That is reverse transcription. And this is halted by Retrover, or AZT. And that means that the virus can't execute its life cycle, and that's good because it means we can control viral replication in cells, and we can help to contain HIV. We can even give it to mothers and help to prevent the spread of HIV to their infants in utero. We can also use it if you think you might have been exposed to HIV and you're put onto drugs such as Retrover to try to prevent actually contracting the virus, allowing it to establish itself in your body. So this is a really, really, really important drug out there, and it is an example of a base analog. It's an analog for the nucleotide thymidine or thymine, the more modern name. If you're interested in war history, one of the first chemical warfare agents was something called mustard gas. And mustard gas is the chemical methyl methanesulfonate. Just gets nicknamed mustard gas. It was first used by the Germans in 1917 in World War I. It is still used as a chemical warfare agent today. For example, the Iraqis, Saddam Hussein was thought or suspected to have used mustard gas on some towns that he was accused of attacking, and it was used in the first Gulf War, and it's still a concern from the point of view of terrorist activities. Anyway, it is an example of something called an alkylating agent. Okay, so to alkylate something means that you add an alkyl group, that's a functional group, for example, CH3 or CH3CH2. The action of an alkylating agent called ethyl methane sulfonate, or EMS, is shown in this figure. And here we see we are going from guanine to 6-ethylguanine. We have added C2H5, that's this. Okay, and when you add that to the environment during DNA synthesis, it will base pair not with cytosine, the way that guanine normally does, but with thymine. So it will induce a nucleotide substitution anywhere where you would have already otherwise had GC base pairing. Intercalating agents, such as ethidium bromide or proflavine, produce mutation by sandwiching themselves, which is what intercalation is, between adjacent bases in DNA. This has the effect of distorting the 3D structure of DNA and causing single nucleotide insertions and or deletions during replication. An example of both is shown in the figure that I'm indicating here. Well-known intercalating agents include proflavine, which is used, or at least historically was used, as an antiseptic compound. Also ethidium bromide, which is used routinely in the lab because it binds so well to DNA and RNA, and it is fluorescent, so it allows us to visualize these nucleic acids in gel chromatography. Or acridine orange, which is a dye that is commonly used to stain nuclei so that you can look at them using a fluorescent microscope. Here up in the top we see a molecule of the intercalating agent. It's wedging itself in, and this is a somewhat random insertion. And because it's wedged itself in, we have a 1 nucleotide insertion at that position after DNA replication occurs. Replication has to occur in order for this to become a mutagenic event. Down here we have a loss or a deletion event occurring, also as a result of the intercalating activity of one of these compounds. Okay, an adduct means a piece of DNA that is covalently bound to a mutagenic chemical. And this figure down here is showing a DNA double helix that's been flattened out, and this yellow area, that's the adduct. It's something that is covalently bound to this, the base on this nucleotide. Okay, the base is in blue, and the yellow is the adduct there, and this is a covalent bond right in this region. And there are many adduct-forming agents that can occur in nature, but also chemicals that are the result of human activity that cause increased levels of adduct formation. Acetaldehydes are produced whenever we smoke cigarettes, and they are also probably produced in our bodies as a side effect of metabolizing ethanol during drinking. Benzoapyrene is produced when we smoke cigarettes or when we char meat on the grill. Heterocyclic amines are also produced when we cook meat. These are just a few examples of adduct-forming agents. When you have adducts in the DNA, the result is a distortion in the double helix, and that can result in base pair loss or insertion, or occasionally it can result in just incorrect base pairing during replication. It disrupts the normal processes of the DNA and triggers point mutations. All right, so radiation is an example of a physical mutagenic agent. The previous discussion in this lecture is focused on chemical mutagenic agents. Hermann Mueller, back in 1927, demonstrated that mutations in fruit flies could be induced by X-rays, and that was in fact one of the first well-documented studies of the genetic damage caused by radiation. Radiation is electromagnetic energy, and electromagnetic energy travels in waves, as you're aware of, and so we describe electromagnetic radiation by referring to the length of the wavelengths in nanometers typically. The visible spectrum spans from about 750 to 380 nanometers, so it's in here. That's not mutagenic. Where we worry about mutation is where we have short wavelengths. So as we travel over to the right-hand side of this particular cartoon, we're getting into shorter and shorter wavelengths, which means higher and higher energy levels. And so it's over on this side of the spectrum that we see danger in the form of mutation possibility due to radiation exposure. So mostly we talk about radiation exposure inducing mutations when it is ionizing or UV radiation. So UV occurs here. We get a lot of that just from the sun. You get a lot more of it if you expose yourself to UV light in tanning beds, which is not a very good idea. And then ionizing radiation, which means you have such high levels of energy that it's actually knocking electrons out of chemicals and generating free radicals. And so X-rays and gamma and cosmic rays are all examples of ionizing radiation. That's kind of the level of radiation that you would experience if you were in any kind of nuclear event. Very, very dangerous. Causes point mutations, causes widespread chromosomal damage, will be lethal very, very quickly. The classic example of damage to DNA that occurs as a result of exposure to ultraviolet radiation is the formation of what are called thymine dimers. Here we have two thymines back to back, so imagine that they are placed in a strand of DNA and they are side by side, and we're just seeing the thymine bases there, not the rest of the nucleotide. UV radiation exposure occurs, and look what happens. We have the formation of covalent bonds here, and the result is a structure that is called a thymine dimer, shown here and here. And that thymine dimer, when you look at it in the context of the three-dimensional double helix down here on the bottom, the thymine dimer leads to a distortion in the double helix. And that distortion in the double helix can be repaired, and we'll talk about something called photolyase in the next portion of this lecture. But if it's not repaired, it can actually make the DNA very fragile there, and the repair mechanisms that kick in to deal with that fragile DNA can result in mutation events.